in
especially
listen-only
for
mode.
we old-timers, because I have absolutely no idea what an omic is or what an omic is doing at Providence. So we're going to learn real fast. I want to remind you all, if you're doing this for CME, docs and osteopaths and stuff, you have to log on to the website and push all the little buttons every day to get your credits. I do it about once every 6 weeks. They'll let you accumulate them. But we have today Dr. Bifulco, who is a surgical pathologist with subspecialty training and expertise in molecular genetic pathology and hematopathology. He is currently the Director of Molecular Pathology and Pathology Informatics at Providence Regional Laboratory and Director of Translational Molecular Pathology at the Earl A. Childs Research Institute in Portland. He has served at the University of Florida, Yale University, completed fellowships in oncological surgical pathology and molecular genetic pathology. This is really taxing me, folks. At Memorial Sloan Kettering Cancer Center and hematopathology at Yale. His current work focuses on supporting the characterization of tumors, genomics features through next-generation sequencing, and of the tumor's immune microenvironment via immunohistochemical and image analysis techniques.
Thank you for the kind introduction. Is the sound okay? Or? Yep. Okay. Great. Wonderful. So thank you for the kind introduction. Thank you for having me here in Missoula. It's an honor and privilege to be here. So that's the title, as you heard, and omics is a little bit funny term. We will really talk about genomics. There's way more going on, but genomics is going to be really the focus of the presentation. This is me. You heard way too much about me already. That's a landscape in Portland. It doesn't include the city, as you can see, but it's a beautiful place. Not as beautiful as Missoula, probably, but close to it. I have no conflicts of interest to disclose about this presentation. So the aims for the talk are going to be to give you a little bit of an introduction to what's going on in the field of genomics in general. And try to share with you what I believe are going to be changes which are going to impact clinical practice and research as well. At the same time, I would like also to give you a few vignettes or a few slides about the Providence personalized medicine program, which is based on genomics. So what you see here is something you're probably very familiar with, which is based on the discovery by Crick and Watson of the double helix. So it's a flow of information which goes from the DNA to proteins. And basically we are made of proteins. So all the things we are made of are actually encoded in these 4 base nucleotides at the level of the DNA. And although the dogma is quote-unquote a dogma, so things are way more complex than they're shown in this cartoon. Information is actually flowing in different ways. I think it has been a very important and useful concept and is overall still very applicable. How does it translate really in pictures? So each of us has 3 billions of nucleotides, the same ones, the 4 ones you saw before, and they basically make a huge book. We actually have more than that. We have a diploid genome, so we have 2 copies of that. And again, there's a lot of information which is coding what we are made of. And if we're able to read that, actually, we are able to gain a lot of potential insight in diseases and other things. And so here an example, you have a colon cancer phenotype encoded in those letters. Translating those letters is not easy, though. And if you look at a timeline, which went from the discovery of the double helix by Watson and Crick in the 50s, you will see that it took a way long time before we were able to read that book. You will see there's a gap of almost 20 years. And the first paper, I think, was a paper-based, I think was, I don't remember, it was Nature probably, 20 base pairs being read. And it took a huge investment of energy, sources, etc. And things didn't really change dramatically until the 90s, I would say. When together with the introduction of automated sequencers and with the start of the Human Genome Project, we had really a game change kind of ability of reading this kind of book. And you can see here that in 2000 the whole human genome was sequenced, which was a major breakthrough. And then there's a further acceleration, very quick, in the later portion of in the recent years, if you want, in the last 10 years, with the introduction of next generation sequencing. The next few slides are going to be really focused on next generation sequencing. I would like to share with you the principles which underline the technology. So you probably heard about the promise of having a $1,000 genome. This is a simplification, and it doesn't really include the whole picture, but you're very close to that number right now. I think this kind of platform, this particular platform, this slide is coming from 2014 when they were put in the market. And as a consequence of our increased ability to actually read that book, we're now getting very efficient in it, and we have this huge increase in data that have been accumulating over time. You will see here the scale now of data of genomes which have been sequenced is gigantic. And the other feature which is quite remarkable is the fact that the velocity by which we are getting faster and better in doing this keeps increasing constantly. So one easy comparison is the famous Moore's law, which underlines everything we do in electronics. It underlies your iPhone, etc. And basically has to do with the doubling of our computational ability every year. And in genomics, things have been evolved significantly faster than that. And the expectation is that things will evolve even faster. So again, the Moore's law, which you see here, is basically not keeping up with what we expect will be the growth on the genomics side. So quite remarkable. We talked about DNA before as being a book that we can sequence and read, but there's way more than we can actually sequence and read. So there's information at the level of the mRNA, transcripts here, information coded in the methylation of the genome, so epigenetics. There's information in many other fields which still can be translated in a string of characters, if you want, in something that we can read as a book. And we have now the ability to do all that, actually. So not only the DNA, not only the billions of bases I showed you before, but more than that. And this slide, again, highlights the fact that we are able to do way more with the same technology. So the same approach, but again, the ability to read way more information. And this highlights, again, the complexity of what we have. So a lot of things that I'll show you will be simplifications, but there's way more complexity, even at the level of the RNA, for example. But again, we have now the ability to read all those different types of RNAs. The next few slides are going to be a little bit busy, and they will try to underline the fundamental principles by which the technology works. The best way really to get an understanding of how NGS, or next generation sequencing, works, which is a technology which enables our ability to massively read the genome, is really to look at a video. You will find them on YouTube. All the companies have them available. And the cartooning will be a little bit difficult to follow, but what is really highlighted here is that we read fragments. We don't read one chromosome from the beginning to the end, which is a huge piece. We don't read the whole book from the beginning to the end. We have to break the book in many different pieces, and we read little pieces at a time. And the thing that's highlighted in this piece of the slide. The other concept which is very important has to do with the fact we have the ability to read all those little fragments in parallel. So the way that we actually able to read the book very quickly, we break it in pieces and we read each piece in parallel at the same time. We don't have to wait for the first piece to be read and then move to the next little fragment. So massive parallel sequencing is really probably the best description of the technology. How this particular technology works is based on fluorescence, and you can see single nucleotides which are assembled one at a time. I think the next picture will show you really how this looks in real life. This is actually what happens when sequencing is getting done, and each little color is a different nucleotide getting accumulated, and the whole image that you see here is then analyzed and deconvoluted and then transformed in strings, in characters, these characters we were describing before. So this is one of the technologies, the different ways to do this. This is currently the dominant technology. It's the one which is underlying Illumina, which is probably a dominant vendor on the market right now. Different approach here. In this particular case, I think things work actually on a little electronic chip, the same chip which powers your phones or your computers. And the detection of the nucleotide change is actually based on a pH change in a little microwell of the size of a few microns. I showed you before this dramatic exponential increase in our ability to read things. The technology is amenable to being optimized, and there's a lot of work in robotics, etc. And again, huge economical interest in trying to make this work better and faster. And what I've described to you so far actually is becoming outdated very rapidly. There's a third generation actually, which is based on a different concept, which promises to be another game changer. So even faster and more efficient than what I described before. It goes under the name of the third generation NGS, next generation sequencing. And again, there's a variety of vendors, a variety of different technologies. But here the concept, instead of having this parallel sequencing of little fragments, is actually to be able to read live a single molecule of DNA. So instead of having a complicated chemistry, these huge amplifications, a huge parallel process, you would be able to read a single molecule at a time. Again, small fragments, but a single molecule at a time, because the sensitivity of the technology is getting so powerful. And in this particular case, I think is a platform which is based on streaming this DNA molecule, a single chain of the DNA, through a little nanopore structure and detecting an electrical change when each base pair goes through the channel. And you can see that this particular company, for example, is called Nanopore, promises to miniaturize things dramatically. So what you saw before, you saw the huge machine with big robots, etc. It's very likely that the technology and the evolution of technology is going to push towards platforms which are way smaller, more portable, and more accessible. And this particular one runs as a USB stick on your laptop. So what I'm showing you, is already on the market, but it's not working yet perfectly. It hasn't defeated yet the big machines, but it's very likely it's going to happen. So what are the problems? Well, I mentioned before the fact that we have this big book which we have to read in little fragments. So there are two things which are challenging. One is trying to get a big picture, the big book, out of little fragments which are randomly broken. The other problem has to do with the fact that even if the technology is very good, it's not perfect. So not only the fragments are fragments, but they also have a lot of mistakes. And so we have to try to get back the original text, if you want, from little fragments which contain mistakes. And that can be quite a challenge. It can be a challenge from a computational point of view. It is also a challenge from an interpretation point of view. Even after you have the right piece of text, you still need to understand what the meaning of the text is, right? And I showed you before all the beautiful colon cancer and red and red hair phenotype, etc. But the reality is that our knowledge is not yet up to snuff to have really one-to-one translation of the text. And there's a lot of work going on there. And so you basically, when you're trying to interpret these things, you have to reach out to very different databases, which are available on public sites or private sites. You have to have an understanding of what is underlying all this. And the bottom line and the message I'm trying to convey you that from trying to translate this in the clinical arena, it becomes a pretty difficult process and quite complicated. And so what you see here are workflows which are actually underlying a deployment in the clinical practice with several steps, several databases, several filters before you actually can get to something which is of practical utility. And the reality is that the field has been changing so rapidly. I think we are getting so much better in our ability to generate the data sets that the ability to actually comprehend them has lagged a little bit behind. And so when you actually try to do this work, you end up having to do a lot of work in-house, trying to build that informatic platform that I was describing before as being necessary to get to clinical comprehensible data sets. And so what you see here actually is a software stack that we had to build in-house in Portland, for example, where we had to build from scratch a bunch of platforms which go to the databases, get information, filter it, make all the workflow actually function. And if you were interested in IT, you would probably recognize the symbols. I presume that you're not, so we'll not spend time with them, but basically they are all platforms which are enabling you the development and deployment of software. So I mentioned before the fact that there's you require a lot of work on the software side, but you also require a lot of work in terms of infrastructure. So not only there's a lot of informatic needed for deployment, but there's also a lot of computational power needed for deployment. And that can be quite expensive even in the current times, even with the computers that we have now, which are quite remarkable. You all know that our phone is better than a mainframe a few years ago, but still it can be a huge challenge. And it can be a particular challenge in the healthcare organization kind of environment, which tends to be very conservative for very good reasons. and building that kind of infrastructure can be a major problem. And I think that's what the slide here is really trying to tell you. This is a medium-sized, almost small-sized platform now, and you can see that the throughput is quite huge. We're talking about terabytes of data. So very different from what you use usually in healthcare, where we do a test and we get a little, yes, no answer, 1 bit. Way larger than that. And so one way to actually approach this is actually to use the cloud. So the cloud is infrastructure which enables almost everything you do on the web right now, including the streaming of movies on Netflix. But the cloud is actually also a very good computational environment for this kind of work. One of the great features of the cloud is that it has the ability to burst. So if you have a lot of computational work which needs to happen with very short time frames, it becomes very easy to scale it up in that kind of environment. And you can then just shut down the whole infrastructure very quickly once the infrastructure is not needed anymore, which is why you see it so widely adopted for a lot of problems. But it's also a great platform for genomics as well. And the government has now sponsored 3 clouds. One is from the NCI, one is from the ISP, Institute of System Biology, the other one is from the Broad Institute. They're all sponsored by the government. A 4th big cloud is the one offered by Illumina itself. And I think that's most likely the environment where we're going to do our computational processes as well. Lastly, even after you have read all the information, you're comfortable about the meaning of that biologically, you're able to say this gene is predisposed to cancer, this patient has the gene which is predisposed to cancer. There comes another level of complexity which has to do with the integration of this kind of information with our conventional tools of clinical work, like EPIC, Cerner, whatever you're using in the lab. And that is a big hassle as well. I think it's a very important hassle because you want to make sure that information is consumable, that it actually drives patient care efficiently, and that it actually works well in the workflows of routine patient care. The other component of this also has to do with the aggregation of the data sets. If you deploy this at scale, you end up having data sets which really involve large populations potentially. And there's a lot of power there because you can discover new relationships. You can actually potentially find out about new drug sensitivities or get information about outcomes, etc. And so in particular in our place, we are currently at the SCI, we have currently deployed a platform that's called SciAps, which does all the things that I described you before. It basically provides a friendly portal for the clinicians to actually access this kind of information, at the same time enable the integration of the information with the EMR, with the clinical data sets, and at the same time enable the aggregation of the data sets for larger purposes. This platform has also been selected by ASCO as their platform for their personalized medicine programs as well. So we're quite happy with that, and we hope that we'll be able to extend the platform to the rest of the Providence system very soon. So I gave you a lot of technical introduction right now, and probably I phased you. But what is the meaning of all this? Well, I guess the meaning of this is really that we hope that the knowledge that we acquire on the genomes will enable us to better cure diseases fundamentally. And you can see here, this is a picture from a cover from Science. The picture I showed you before with all the colors actually is coming from here, as you can probably infer, and this was published in 2015 as a review just focused on genomics. Again, underlying the importance of this right now. And the scope of this is really covering multiple areas of medicine. It goes from inherited diseases, infectious diseases. I would say there's not a single field of medicine which is not going to impact by this. You can use this for diagnostics in multiple areas, and I think we will see transformational changes in all of them. But the one I'm going to focus today is really going to be cancer care, because that's the area that I have a personal interest, and that's also the area where we're going to do our first push at a system level. That's where we are really going to be focused initially. And what underlines the importance of genomics here really has to do with the paradigm, if you want, of the hallmarks of cancer. Is a paper by Hannah and Weinberg, I think a few years ago, very much quoted, where basically our understanding of cancer is that to acquire the malignant phenotype, which basically enables cells to grow independently and then invade tissue, and finally to metastasize, and often, unfortunately, to the demise of patients, you usually have a need for overcoming several fundamental restraints. And so you can see here evading apoptosis, which is cell death, is one of those. having the ability to sustain angiogenesis on your own is one of those. Being able to replicate limitlessly is another one, and there's a bunch of those as well. And each of those steps, we believe, has a fundamental genetic mechanism underlying it. So each of those will have a mutation, a change in the genome, which underlines it. And that is really what is behind our current understanding of cancer, or current model of cancer, where we see cancer as being a genetic disease. What you're seeing here is Vogelstein's model of progression in colon cancer is a well-accepted, basically the framework for our current understanding of many things, where you have initial mutations which initiate the transformation into a malignant phenotype, and then the accumulation of those other events I described before, like invasion, etc., each of them links to a specific mutation. And you can see that once you have multiple of those, you really end up having a transformed phenotype. Initially, you just have a growth of an adenoma, but then you have something which is able to invade tissue, and unfortunately then go to the lymph nodes or metastasize, etc. So this is all nice and great. We have an understanding of the biology, but what else? Well, those changes that I showed before in the scheme of being hallmarks of cancer are potentially targetable. So each of them basically has a change at the book level and the DNA, but then becomes a protein, right? The protein which then does a job and gives you this kind of aggressive phenotype if you want. Well, each of those is potentially be a target. And you can develop small targets, small molecules, or different approaches to try to revert that or block that. And that is the concept behind the targeted therapies, where you have actually a change at the nucleotide level in the DNA, and that gets translated in the protein, which is altered, and then you can potentially block that mechanism. And I think that the last probably 10 years have seen the introduction of targeted therapies in clinical medicine and oncology. Particularly, you see now that lung cancer, they play a major role, melanoma as well, breast cancer as well. Multiple tumor types you will see are now becoming routine targets for target therapies. Apologize for repetition in clinical practice. And I think you're all very familiar with what I'm showing you here, or at least the people who are involved in clinical work are very familiar with what I'm showing you here. And this is just one example in case you are not, but basically we have here mutation, and this is melanoma, you have mutation in BRAF, and there's a specific drug which is able to block that mechanism. This is on the other side of the slide, you will see the conventional testing mechanism, which is basically you look at the specific mutation only at one at a time. And what happens when you actually look at the big picture, you will see that in reality, cancer has multiple pathways. It's not just one single mutation, and there's a multitude of targets which are actually potentially targetable. So not a single gene, many genes potentially altered at the same time, multiple targets being available. And you will see also that drug companies have a good understanding and very much an interest in this, and have developed a variety of new novel approaches to hit each of those steps. So now we have a picture where we have diseases which have multiple alterations and potentially multiple drugs available at the same time targeting each of those, which really makes the case for NGS testing. So going away from the single gene testing that I mentioned before, where you test the single, specific disease, a single mutation, but you really want to move to an approach where you actually get the whole picture at the same time. Multiplex testing if you want. And so what I'm showing you here are actually the guidelines for lung cancer testing. They used to be test one gene at a time, find the next one if the patient progresses. But here they explicitly mentioned for the first time next generation sequencing as a standard of practice. And I think what you're seeing here in lung cancer is a model which is going to be repeated in every single different tumor type. You will see it applied to colon cancer, you will see it applied to breast cancer, you will see it applied in leukemias, lymphomas, etc. Basically, getting the whole picture in one shot is way more convenient than getting one piece of information at a time. So how do these targeted therapies work? Well, we talked about before about the targets and the small molecule blocking it, and they can induce dramatic remissions. And so what I'm showing you here is a patient with melanoma. You can see, I think he got an inhibitor of BRAF. You can see the dramatic response. These drugs have been around for now a bunch of years. One underlying limitation of this, though, is the fact that you end up very often developing resistance. So they induce dramatic remissions, they dramatically improve patient care, but they don't cure cancer. And I think that has been true for every single one of these targeted therapies, unfortunately, in the sense that their ability to actually block the underlying mechanism is temporary. You often have emergence of resistance in the form of alternative pathways or of mutations which then block the action of the drug. And there's not only those mechanisms I mentioned before, there's also a very fundamental problem which has to do with clonal heterogeneity. So cancer is basically characterized by all those alterations I mentioned before, genetic changes. But one of the features also is the fact that it's very unstable. And that you end up having very different cell population, each one with a different set of mutations, which is really what this picture is trying to show you. So you have an initial event, initial founder mutation, but then you end up having different populations at the end, each one with a different set of features. And that also explains why our target therapies are not so efficient. We are targeting one of these populations. We're not targeting all at the same time. And so as soon as you kill one of those clones, if you want, you will have to deal with the other clones. And that's what happens in real life in our patients right now with target therapies. And not only we have a difference of clonal heterogeneity, if you want, in the tumor, but we also have clonal heterogeneity in between different metastases in the primary tumor. So each MET will have its own subset of clones, and each MET will be different from each other. And they will all be different from the primary because you have this kind of evolutionary Darwinian process, if you want, and which basically brings in very, very hard and challenging limitations to our current approach of a single target at a time. So what are the ways to actually address that? Well, we talked about sequencing. We like to sequence the tumors. We talked about the diagnostics. We like to drive targeted therapies. But one way to potentially address this is by actually looking at what happens in the blood. So the tumor cells have the mutations, they have all these little changes that you can actually detect at the blood level due to the fact that tumor DNA is actually available in the environment. Is quite resistant. So you actually can sequence the blood and then you can actually infer what is going on in the patient. And I think that is quite a promising approach where you give the patient a targeted therapy, you see what changes in that environment, you see what a new emerging clone is, and then you give a new targeted therapy trying to address that. And so that's one of the potential promises for having transforming cancer in some kind of chronic disease where we keep targeting new specific mutations over time. And obviously it has huge advantages because it's non-invasive, it's easily accessible, etc. And so there's a lot of promise there and huge investments in this area. Another approach, though, has to do with a completely different kind of mindset, which has to do with immunotherapies. I think this is the cover of the year. This was in 2000, I believe, 13, so now a few years old. But immunotherapies have been around for a while. They had sporadic, dramatic remissions, but really no substantial impact on patient care. But things changed recently. I think people here are very familiar with the power of these right now. They're becoming really mainstream, and one of the reasons why I believe they're so powerful has to do with the fact that the immune system is built for dealing with heterogeneity. If you think about viral or bacterial kind of populations, they tend to be heterogeneous, mutating all the time as well. And the immune system is really built for dealing with that. The other source of strength, I believe, has to do with the fact that the immune system is a self-reinforcing platform. So what I'm trying to say here is that the more tumor cells the immune system is killing, the more antigens get exposed to the immune system, and the more you have a kind of positive feedback mechanism by which things get stronger and stronger. So this is one of the avenues where things can change. And I think it's not, again, a few years old now. Walter is actually the director of the Cancer Center in Providence, in Portland. And you can see that in his presentation at ASCO, he highlighted the fact that immunotherapy tends to be a little bit lagging compared to target therapies, where the effect is very dramatic in the beginning. You have these dramatic regressions. But if you look at long-term, actually, you end up having this long tail of response, which is often absent in targeted therapies, and which is very promising from a survival point of view, because that's what it's translating in. So basically, potentially, actually, here we have the ability in the future to cure metastatic disease, which is something very novel. And I think the picture you saw before was with the first immunotherapy. This was melanoma, which was based on EP, but you can see now that we have PD-1 pathway inhibitors, and I will not go through much into detail of how they work, but the fundamental mechanism by which they work is by unleashing the immune system. You can combine them and you can get way more significant response rates, and I think now the response rate in melanoma is around Metastatic melanoma is around 40%. And I think we see significant response rates now also with lung cancer, kidney cancer, and other tumor types where they're around 15%. So a dramatic change from before. Things work very differently from what you saw before. You saw before a picture of a patient with a dramatic response, immediate. With immunotherapy, the effect is a little bit different. Things tend to actually to not respond immediately, but respond over time. You often have a freer effect, most likely related to the fact that you have the activation of the immune system and actually infiltration of the tumor by immune cells. So very different kind of time frame. But what is really remarkable of those, again, is the fact that often these kind of responses are long-lasting. So what is the role of genomics now in the era of immunotherapies, which I think is what's going to happen in the next few years? I think we will see a transition from small target therapies to the use of biologicals and to the application of immunotherapies. You can see that being recognized by the press, I think it's coming from The Economist as being a big player as well. I think there are strong links between genomics and immunotherapy. And they fundamentally have to do with the fact that all those changes I told you before are potential targets for the immune system. There's something new the immune system has never encountered before, each of them. And so what this slide is trying to highlight actually is the number of changes that you have in tumors. And so tumors have very different mutational loads. The book that we described before can have different levels of changes compared to the normal book. And some tumor types tend to have a lot of those. And interestingly enough, if you look at on the right side of the spectrum here, melanoma, which has the highest number of changes, is a little bit a poster child for immunotherapies. And the reasoning most likely has to do with the fact that it is so loaded of mutations, which makes it an easy target for the immune system. The same is true for other tumor types, which now we have pretty good evidence that immunotherapy is working. And so lung cancer is also here high up as well. And so this is the first link, if you want, between genomics and immunotherapies. By having an understanding of the mutational load, you can actually predict who is going to respond to that kind of therapy. There's more than that. I think there's now evidence that some of these mutations actually have some links with potential epitopes, which the immune system is built to recognize. And so we are obviously built to recognize certain infectious pathways, and there's some evidence suggesting that there's a shared common ground between those mutations we see in the tumors and which then are amenable to immunotherapy and some of those infectious kind of pathways as well. And there's a lot of data accumulating very rapidly, I think, in the last few years about the relationship with genomics and immunotherapies. And so here you see two papers. I think this one here is in melanoma. I think a couple of years coming from the Chan group with Morris and Kettering. This other paper also from the same group is about lung cancer. In this particular example, I think what they're trying to show you is that survival with immunotherapy is way higher if you have certain changes. Interestingly enough, those changes are correlated with smoking. So that mutation here I'm showing you here is actually a mutation which is induced by smoking, and the patients which have smokers actually tend to do better in this particular subset of cases when applied to immunotherapies. The slide is very busy, and I will not spend too much time about it, but again, the same concept. I think the more mutations you have, the better you're targetable with immunotherapies. And this is the most commonly targeted pathway. It's called the PD-1, PD-L1 pathway. It's the most common target for immunotherapy activation. And more of the same. This is a relatively recent paper. One of the authors is one of member of our group is Dr. Chancy, which is named, I think, is somewhere in here. But you will see that in colon cancer, same kind of picture. So colon cancer has a subset. It's called MSI high. Many of you are familiar with it. Basically, there is a problem in mismatch repair genes, and it ends up the colon cancer which have this mismatch repair problems tend to accumulate a lot of noise in the DNA. And what you see in these graphs is basically that patients who have these alterations are way more sensitive to immunotherapies than anybody else. Again, a link between genomics and immunotherapies. There's even more than that, I think. So what we talked so far had to do with generic immunotherapies, where the idea is let's remove the brakes from the immune system that a tumor is produced, and let's try to activate the immune system as much as possible and see what happens. And the immune system doesn't work on its own. But there are other ways where we actually can personalize the immune system. And so in this particular context, I think the idea is to identify the specific mutation I showed you before and build T cells or B cell mechanisms which are specifically recognizing those changes. So for example, we could take a T cell which recognizes the BRAF mutation and expand it and then push it back in the patient and have a very specific, targeted, if you want, immune agent. And they can be extremely powerful. And they're right now being deployed quite commonly in B cell leukemias. BALL is currently a target where there's incredibly promise with this kind of approach, CAR therapies, but they're also getting deployed in solid tumors. And I think you've seen the beginning of that at least. And you can see here a bunch of papers coming from Rosenberg's group at the surgery branch at NCI, where they actually are doing what I described before with success in a few patients. Processes are still very complicated, time-consuming, not clinically deployable in routine practice, but certainly promising and very interesting. So we talked a lot about genomics, and I mentioned before that the presentation is really focused mostly on cancer. But another feature I would like to mention, without going too much into detail in different tumor types, is to the fact that we have potential for really preventing diseases rather than just treating them once they're available. So one example of that is by work done by Mike Snyder, who is the director at Stanford for the Center for Genomics and Personalized Medicine. And so he published a paper, I think, in Cell. This was probably 2 or 3 years ago, where he did anything he could on himself from a genomics perspective. And so he not only got his own genome, but he got any kind of sequencing that is going to be for RNA, et cetera, continuously on a monthly, weekly basis. I don't remember the detail of the paper, over a period of time. And what was interesting was that he was able to detect a small infectious event. He was able to detect it at the genomic level, which then resulted in damage to his pancreas, to the islet cells, and resulted in him having diabetes fundamentally. And he was able to detect that before the time and have very aggressive interventions. So the idea is actually that to deploy this kind of technology in a preventive kind of fashion. And people are really trying to push this quite hard. This is a potential future collaborator of Providence, and also one of the founders of NGS, Next Generation Sequencing. Some of the platforms I showed you before at the very beginning, which were underlying our ability to sequence the whole genome and the whole genome project, the success of that, were actually developed by him. And you can see he's now pushing for a predictive, preventive, personalized, and participatory medicine, P4 medicine, where the idea is to apply all those technologies and really try to change the way we deal with diseases, trying to prevent them before they actually start. And that's Lee Hood at the ISB, the Institute of Systems Biology, and a potential future partner for Providence. What I show you in this slide is that on the foundation of a whole genome, you can then actually build additional data sets with continuous monitoring with, again, the intent of, instead of treating with, dealing with a disease which is full-blown, actually having early interventions and prevent the evolution of those. And so the genome is really the foundation. It will give you predispositions for certain diseases. It will potentially change the way your lifestyle evolves. But then on top of that, you have additional interventions in forms of monitoring. The idea is actually to monitor even physiological things with sensors, et cetera, and then build those huge data sets, put them all together, and try really to personalize completely our approach in a preventive fashion. I think this is getting deployed on a commercial level already, but it's potentially going to be deployed also at the clinical level in institutions as well. Lastly, I wanted to mention that all this movement that you saw before, all the action that you saw, definitely reached the level of the government. The government has been very fundamental institution in this. If you think about the Human Genome Project, it was sponsored by the government. It really was the foundation for everything I've been talking today. But they're getting involved in this as well, and you can see that I think this was the address in 2015 at the beginning. In 2015, there was a launch of a precision medicine program, but most recently, I think this was probably just a few months ago, there was an announcement of the so-called Moonshot program as well, again with the idea of leveraging the power of genomics and really trying to change the landscape of how we treat cancer. And again, a lot of going on at that level. So the last few slides are really about what Providence is doing in this field. We'll try to give you a brief introduction of what our deployment is. This particular slide is very busy. I think it shows you a lot of text basically describing what our lab in Portland is. Right now we have 2 centers which are offering genomics in the system, one in Seattle at the Swedish Cancer Institute and one in Portland. I'm in Portland, so that's what the slide is showing you, but this is a sister lab in Seattle as well. We offer testing already, at least locally, and this slide here describes what our offerings are currently, but we have more going on. I think there are the stuff that we're doing in a research environment only. Some of the stuff is done in a clinical environment only. So current existing offerings, but it's also a program which we hope to expand where we have a wider and I would say standardized approach that we then will make available throughout the whole system. And the way we try to develop this is really being instead of being from the top to the bottom, I think we really try to build this from bottom to top. So we had a lot of interactions with clinicians, consumers. Clinicians are our consumers, quite frankly. And we try to build this around 6 pillars that you can see here. There's been a lot of work going on with each of these pillars. I'm involved in a couple of those. But some of them have to do with the lab, some of them have to do with clinical applications, some have to do with the tumor registry, some will have to do with research, etc. We are really trying to build a platform which best serve the interests of the whole system. So just to give you a brief preview of where we think things are going to go, I mentioned before the SciOps platform, which really enables the clinicians to interact with this in a friendly fashion and integrate with the EMR. And so we hope that we'll be able to expand the platform from out of the initial deployment, which is at SCI, at the Swedish Cancer Institute, but we hope that we'll be able to deploy that to the whole organization very soon. We will offer a very large panel. When I mean panel, I mean that we'll sequencing 200 or more targets, those famous targets that we described before being potential actionable items. So it's going to be a relatively large panel, hopefully available soon. And the idea is that we'll initially deploy this in the Cancer Institutes, but then hopefully be able to extend the deployment to the whole Providence system, which is, as you probably know, becoming very large. I think it just recently almost doubled in size. Lastly, I think we talked a little bit about immunotherapies, about the role of this in immunotherapies. And in that setting, I think the data sets that we need to get to really drive those are larger than the panels, larger than just the targeted therapies. We really need to go to whole exome RNA-seq or more wider approach. And so we have a lot of work going on there, and we hope that we'll be able to transition out of this initial phase, which is targeted therapy related and driven, to something which is more comprehensive. And so that's going to be our next hope. Lastly, before I let you go, I just wanted to mention that there are additional major changes driven by genomics on the horizon. They really have to do with the fact that not too long ago, people discovered new ways to actually cut and paste the genome. So remember we talked about the book with letters and changes being changed. It is possible actually to take that book which has the changes, it's not the book that you would like to have, to modify it and go back to what you like to have. And it used to be a very complicated process. So actually manipulating the genome and doing what is called gene therapy, if you want, used to be something very difficult. But it's getting actually extremely easy. And that has to do with work, which is interesting enough, has to do with the immune system of bacteria. Bacteria have developed their own ability to cut and paste genomes to defend themselves from viruses. I think something like 20 years ago, people really got into this and discovered how it works. It's called CRISPR-Cas9, and I will not tell you exactly what it stands because it stands for a lot of complicated words, but it's basically the immune system of the bacteria which defends the bacteria from viruses. And by using and leveraging this system, actually, you can now cut, paste, and modify genomes extremely efficiently, to the point, actually, that there are moral and ethical implications. We are getting so good at it, potentially, that we can actually alter the germline of people. And you can see that there's a huge moral and potential ethical problems related to that, and people are almost suggesting some kind of moratorium for the technology because of those potential huge implications. But I'm pretty sure, I'm pretty convinced that you will see the application of this in clinical practice, at least at a somatic level. So you will see changes in tumor cells, not in the germline, hopefully very soon. And this is a review by Eric Lander from the Broad Institute going on the topic. The topic is very controversial, by the way. I think there's huge financial interest in this. People have been arguing a lot about who owns what and who actually has the intellectual property for the development of technology. Lastly, so that's where hopefully we'll go. I think I have many of the defects that you see here in the picture. So only I'm lacking. I have this problem here, maybe some others as well. But potentially in the future, those will be addressable and things will be hopefully different. So I think I'm reaching the end of the presentation. There's a lot of people I want to thank. Walter Orba was in some of the slides, and Thomas Brown. They've been the directors of the Initiative for Providence. The clinical directors, Calvin Harrison and Brian Wilkinson, are here in the room, I think. They've been the administrative directors of the genomics effort for the system. And there's a lot of other people which have been working on this very actively, trying to bring this to fruition for Providence. So there's a lot of work behind what I just showed you. And lastly, I want to thank you for your attention, and hopefully have a few minutes for questions if we didn't go over time. And I lost track of time.
I I had no idea the field had gotten this advanced. Thank you for updating us. I have two questions, unrelated. There's some evidence that the biogenome in the GI tract has some impact on cancer. Is that related to this last part you were talking about with the?
Yeah. So yes and no. You're absolutely right. I think that there's very strong evidence actually that the bacterial populations in the gut have a dramatic impact on how people respond to therapies. I think there's some evidence that antibiotics, for example, can dramatically impair your response to chemotherapy, even conventional therapies. So there's a lot of stuff happening in the field of sequencing bugs, quite honestly. So yes, and that could be is potentially also one of the drivers of the immunotherapy as well. So if you think about what we are really exposed in terms of environment, we have an exposure to the environment through the skin, through the respiratory tract, but our biggest exposure to the environment has to do with our GI tract, which actually has a huge surface and has an incredibly large bacterial population. And the immune system is monitoring that side as well constantly. So you're absolutely right. There's a dramatic evolution there. I didn't talk too much about that. There's potential links for conventional therapies, for immunotherapies. I don't think too much for targeted therapies, but certainly for those two areas. And I think we'll see very rapid changes there too. One of those slides that I showed you also, I want to mention the ones about the idea of monitoring has to do with the same concept. So part of that approach has to do with sequencing the gut and to be routinely and make sure that actually we have an understanding of how that evolves and how that interacts with the health of people. The part of the immune system was actually a little bit of the bacterial immune system, that the rearrangements, the ability to modify and cut and paste was actually a little bit different. It really had to do with the immune system of the bacteria themselves. But yeah, so that's very important and great topics.
My second question comes from the ethicist in me. I can easily understand how this is very valuable work once someone has cancer. But the P4 concept is very troubling to me. This is incredibly resource rich, and it strikes me as being a further step in increasing the inequality of healthcare.
So I would agree with you. There are aspects of it which are concerning. First of all, it's not a proven concept. There's no evidence yet that it translates in cost-efficient, better care. There's no evidence yet that it really changes outcomes. So that has to be accrued before we can actually go for it. I do agree also that currently, at least, the deployment of that kind of concept is really restricted to people which can afford to pay for it. Having said that, I could argue with you, and that is true for many things that we do. So often we start deploying them to the people who can afford them. And then you have this kind of process by which mass production, et cetera, that makes it available to everybody. So I fully agree with you. I do think that first we need to prove that the concept is valid. Second, I think hopefully we'll see if the concept is valid indeed, and I'm not sure yet about it. I would expect that we'll see economy of scale, and that everything that I showed you before will become so cheap that actually it will be amenable to be implemented everywhere. The last thing I want to mention has to do with privacy also. You could argue that, why should everybody of us be monitored continuously, having constant interventions, which some of us may feel are invasive of our personal privacy ability to make choices. And you can see also links to insurance providers and other things which are very troublesome as well. So the picture is complex. I fully agree with you. There are some ethical issues which will need to be addressed. And again, I think we don't have yet a proof that this is really able to impact care yet. Hopefully we'll have some answer in the next few years. Okay, thank you.